Hello everyone. This class will be a introduction to the renal system. Objectives of this class would be to list the function of kidney, draw schematic diagram of nephron, identify parts of nephron and their function, correlate histological modification at different segments of nephron with their specific functions, list the differences between cortical and juxtamedullary nephrons, outline ultrastructure of glomerulocapsular filtration barrier, list the functions of glomerular mesangial cells, name the components of juxtaglomerular apparatus, and describe the function and structure, and elucidate the steps of activation of renin-angiotensin system, enumerate functions of angiotensin 2, and appreciate its clinical importance. The kidney plays a vital role in homeostatic functions of body. Therefore, impairment of kidney function leads to many homeostatic abnormalities. Extracellular fluid compartment is the interface between external and internal milieus of living creatures. Water, minerals, nutrients, and gases pass through the ECF before entering the cellular compartment, and cellular waste products pass through ECF before being excreted from the body. Hence, maintenance of ECF volume and composition, the, the aim of many homeostatic mechanisms, is vital for the organ functions. The kidney plays an important and principal role in this homeostasis. In clinical practice, biochemical investigations are important weapons of clinical diagnostic amartami. These routine and special investigation for medical or surgical wards and clinics are generally meant to reveal kidney function and dysfunctions, directly or indirectly. Medical students, interns, residents, and consultants need to learn more of kidney physiology to understand and interpret the reports of these investigations. Moreover, understanding renal physiology helps physicians learn the pathophysiology of renal diseases and plan the management strategies of these disorders. Functions of kidney. Urine formation and excretion of waste product is the primary function of the kidney, and they help in excrete waste products of the metabolism of the body, such as urea, creatinine, uric acid, etc., which are dissolved in urine. They help in the regulation of ECF volume. Kidneys play an important role in regulation of ECF volume of the body. They help in regulation of the blood pressure. Kidneys contribute mainly to long-term regulation of arterial volume and pressure. They help in regulation of electrolyte and the composition of the body fluids. Composition of the electrolytes in ECF is mainly the function of the kidney. They help in acid-base balance. Kidneys contribute significantly to acid-base balance by controlling bicarbonate excretion and H plus secretion. By controlling NaCl and water reabsorption, kidneys control plasma osmolality. Erythropoietin, the major regulator of erythropoiesis, is secreted from the interstitial cells of the peritubular capillary bed of kidney. So thus they have a role in regulation of erythropoiesis. Kidneys secrete thromboxane A2 and prostaglandins in addition to the secretion of erythropoietin, so they have other endocrine functions. The kidneys are not the primary site of gluconeogenesis. In starvation, synthesis of glucose from non-carbohydrate source, especially from glutamine, occurs in this organ. So they're also involved in gluconeogenesis. This is the functional anatomy. Kidneys are primary organs for urinary system that also includes ureters, urinary bladder, and urethra. Kidneys are bean-shaped, paired retroperitoneal organs present in the posterior part of the abdomen, just above the waist, between the last thoracic and lumbar vertebrae. The size of each kidney in adult is roughly about the size of one's fist, that is 11 centimeter in length and 6 centimeter in width. and the weight is about 150 grams. Kidneys are covered by a thick capsule which is called as renal capsule. The medial border of kidney is concave and the center of the concavity is called as renal hilus, from which the ureter comes out of the organ. So this you can see the pair of kidneys which gives up the ureters which are going to come to the bladder, and the urine is stored in urinary bladder. If you see the internal structure of the kidney, when kidney is cut vertically into two halves, two distinct regions are seen, outer cortex and inner medulla. The renal cortex is granular appearance and contains all glomeruli, convoluted tubule, and cortical portion of the collecting ducts. The renal medulla is striated in appearance due to the presence of loop of Henle, medullary portion of the collecting duct, and blood vessels that are arranged in parallel. So these are the cut section of the kidney where you can see the cortex and the medulla, and you can see the renal papillae. So again you can see the major calyx, minor calyx, which opens into renal pelvis and ultimately into the ureter. Nephron. Nephron is the basic structure and functional unit of kidney. The nephron are hollow tubes through which the blood is filtered and modified finally to form urine. A nephron consists of renal corpuscle, proximal convoluted tubule, which is called as PCT, loop of Henle, distal convoluted tubule, that is DCT, and collecting duct system. Each segment of nephron consists of a single layer of epithelial cells placed on the basement membrane that are modified to form specific transport function suitable for that segment. So this is a structure of the nephron where you can see uh, the different parts of the nephron, that is the glomerulus with the Bowman's capsule, uh, with totally it is called as renal corpuscle, where you can see the efferent arterioles and afferent arterioles. Then you have a proximal convoluted tubule, the loop of Henle, distal convoluted tubule, connecting duct, and the collecting duct. Renal corpuscle. The renal corpuscle, also called as Bowman's capsule, contains the glomerulus, a tuft of capillaries. Bowman's capsule filters the blood from glomerular capillaries from the ultrafiltrate, which is delivered into the proximal tubule. Proximal tubule, this is the initial segment of the nephron and hence called proximal tubule. As the most part of it is coiled, the proximal tubule is also called as proximal convoluted tubule. It ends in a small straight segment that continues to form loop of Henle. The proximal tubule is about 15 mm long and 40 micrometers in diameter. Proximal tubule recovers from most of the filtrate for its considerable capacity of reabsorption. Loop of Henle is the continuation of pars recta of proximal tubule. It consists of descending and ascending limbs. The descending limb is a thin throughout all the nephrons, whereas the ascending limb has a thick and thin portion of thick and thick portion in a typical juxtamedullary nephron. Thus, loop of Henle in juxtamedullary neurons has three parts: thin ascending, thin ascend, thin descending, thin ascending, and thick ascending limbs. In cortical nephron, a thin ascending part of the loop of Henle is almost absent. The total length of loop of Henle varies from 14 to 26 millimeters depending on the type of nephron. The thin limb is about 2 to 14 millimeters long, and thick ascending limb is about 12 millimeters long. About 15% of the filtrate is reabsorbed in loop of Henle. The loop of Henle of juxtamedullary nephrons plays an important role in countercurrent mechanism of urine concentration. Distal converted tubule. The distal converted tubule begins immediately after the macula densa. It is 5 mm in length. They have extensive infoldings of basolateral membrane of cells that contains abundant mitochondria. The luminal surface of the cell has less infoldings. Though the DCT is relatively impermeable to water, hormones like aldosterone and ADH facilitate its absorption in this segment. Connecting segment. The DCT empties into the collecting duct through the connecting segment or tubule. This is a small and relatively straight tube with morphological and transport characteristics similar to that of collecting duct. And collecting duct, it the DCT is usually empty into the collecting duct. The collecting duct is 20 millimeters long. It passes through the cortex and medulla to empty into pelvis of the kidney at the apex of medullary pyramids. Thus, it is divided into two parts, the cortical and medullary portions. Medullary portion of the collecting duct is further divided into outer medullary and inner medullary parts. The epithelial cells of collecting duct are modified to participate in the transport of ions and water. Water permeability is controlled mainly by ADH, whereas sodium transport E is controlled by aldosterone. Histological modification are as below. If you see epithelial cells, they are cuboidal with minimal foldings of basolateral and apical membrane. Mitochondria are few in number. The epithelium of collecting ducts, there are two types, principal cells, that is P cells, and intercalated cells, that is I cells. P cells are more number than I cells. P cells have a moderate basolateral infoldings and few mitochondria. They are involved in sodium reabsorption and ADH induced water reabsorption. Whereas I cells, they contain more microvilli, more basolateral infoldings and more mitochondria. They are involved in acid secretion and bicarbonate transport. Few I cells also present in DCT. Secretory cells of kidney. Secretory or endocrine cells from kidney are mainly of two types, juxtaglomerular cells and interstitial cells. Juxtaglomerular cells or JG cells, they secrete renin that activates renin-angiotensin system. The interstitial cells, which are called as ISLs, there are two types, the cortical and medullary interstitial cells. Cortical interstitial cells are two types, phagocytic and fibroblast-like cell, where fibroblast-like cells, also called as peritubular interstitial cells, they secrete erythropoietin. Whereas medullary interstitial cells, there are again of two types, type 1 and type 2. Type 1 medullary interstitial, they produce the prostaglandin, especially prostaglandin E2. Okay. So you can see the different morphology at the different part of the nephron. Okay. So in proximal convoluted tubule, these cells are cuboidal and you can see more number of villi compared to the cells which are there in the other parts like loop of Henle, DCT, or the collecting duct. Types of nephron. Anatomically, there are three types of nephrons: superficial nephrons, mid-cortical nephrons, and juxtamedullary nephrons. Superficial nephrons, the glomerular capsules of superficial nephrons are located in the superficial layer of the cortex, whereas the mid-cortical nephrons, the renal corpuscles of mid-cortical neurons, are located in the middle of the cortex. And juxtaglomerular nephrons, physiologically, superficial and mid-cortical nephrons are considered as the cortical nephrons due to their similarity in histological morphologies and functions. Cortical nephrons. Renal corpuscle of cortical nephron is located in the superficial region of the cortex. The glomerulus is usually smaller. Loop of Henle of these nephrons is short. The loop of Henle does not have any thin ascending limb. However, the total length of the nephron is almost same as that of juxtamedullary nephron, as density is longer. Efferent arteriole supplying the cortical nephron branches into peritubular capillaries that surround the segments of nephron of its own and the adjacent one. Juxtamedullary nephrons, the renal corpuscles of juxtamedullary nephrons is located in the juxtamedullary region of the cortex, that is, the inner region of the cortex close to the medulla. The glomerulus is usually larger. They have a loop of Henle that extends from deep into the medulla. They also have a thin ascending part of LOH, that is loop of Henle. The efferent arterioles from peritubulary capillaries and a series of vascular loops which are called as vasa recta. So this is the diagram showing the cortical and the juxtamedullary nephrons. Okay, so this is the cortical nephron and this is juxtamedullary nephron. Okay, you can see the loop of Henle, so which is restricted to the cortex, whereas the juxtamedullary cortex, the loop of Henle descends down into the inner medulla. These are some of the difference between cortical and juxtamedullary nephrons. The cortical nephrons, they are located in the superficial region, whereas juxtamedullary, they are in the juxtamedullary region. Cortical nephrons forms 85% of the total and only just 15% are juxtamedullary nephrons. The cortical nephrons are shorter and medullary nephrons are longer because of the long loop of Henle. Length of the thin limb of loop of Henle is shorter and it is longer in juxtamedullary nephrons. The size of glomerulus is small in cortical nephrons, usually large in juxtamedullary nephrons. The tubular function of cortical nephron is filtration and reabsorption, whereas juxtamedullary nephrons, they are involved in filtration, reabsorption and creation of medullary osmolar gradient. The rate of filtration is slow in cortical nephrons, high in medullary nephrons. Efferent arteriole forms peritubular capillaries, and in the juxtamedullary with the peritubulary capillaries, they also form vasa recta. Vasa recta is absent in cortical nephrons, they are present in medullary nephrons. And juxtamedullary nephrons, they are more important for concentration of urine. The renin content is more in case of cortical nephrons, less in case of juxtamedullary nephron. The main function of cortical nephron is urine formation and excretion of the waste products, whereas the important function of juxtamedullary nephron is to form urine, or rather urine concentration. If you see the ultrastructure of the renal corpuscle, it consists of Bowman's capsule that contains glomerulus and mesangium. This executes the first step of urine formation. The formation of urine involves three major steps, that is ultrafiltration, tubular reabsorption and tubular secretion. Ultrafiltration is the first step in urine formation, occurs in the renal corpuscle, in which plasma is differentially filtrated to form tubular fluid. This is the initial and important step of urine formation, as it considerably contributes to the volume and composition of urine. Glomerulus, a tuft of capillaries located in the initial dilated and cup-like portion of the tubule, and which is called as Bowman's capsule. The glomerulus is formed by the capillaries that arise from afferent arterioles. The glomerular capillaries drain into efferent arterioles. The glomerular capillaries are only capillaries in the body that starts with the arterioles and end in arterioles. The capillaries are covered by epithelial cells called podocytes. Bowman's capsule. Bowman's capsule is the initial cup-like dilated portion of the nephron that accommodates glomerulus. The capsule has two layers, visceral layer and parietal layer. Visceral layer that remains in close contact with the glomerulus and parietal layer that forms the outer portion of the capsule. The podocytes form the visceral layer of Bowman's capsule. The parietal layer of the Bowman's capsule is formed by the parietal epithelial cells, and the space between visceral and parietal layer is called as Bowman's space. Filtration barrier. The endothelial cells of glomerular capillaries are covered by basement membrane. Podocytes surround this basement membrane. The capillary endothelial cells, basement membrane, and food process of podocytes form the filtration barrier through which the blood from the glomerular capillaries is filtered uh, to form the tubular fluid. Okay, so this is the structure of the renal corpuscle, which shows the afferent and efferent arterioles, which is going to form the tuft of the vessels, and this is the Bowman's capsule. And in between, you have visceral layer and the parietal layer. Visceral layer is going to surround the arterioles, and parietal layer is going to form the Bowman's capsule. And in between, it is called as Bowman's space. Capillary endothelium of the glomerular capillary is fenestrated type as it contains fenestra, that is pores or windows. These pores are large in diameter, that is up to 50 to 100 nanometers in diameter, and therefore they freely allow passage of large molecules through them. This allows water and small solids like sodium, urea, and glucose to pass through the capillary endothelium. Endothelial cells are negatively charged due to the presence of anionic glycoproteins on their surface. The proteins in the plasma also anionic. Therefore, protein molecules are not easily filtered through the filtration barrier, and hence normally protein is absent in urine. Basement membrane. The basement membrane is made up of mesh work of fine fibrils uh, entrenched in a gel-like matrix. It is an important barrier for proteins as the matrix contains collagen, laminin, fibronectin, and other negatively charged proteins. Food process of podocytes. The podocytes, that is, food cells, uh, form the third layer of the inner layer of filtration barrier. Podocytes constitutes visceral layer of Bowman's capsule, and they are epithelial cells. Their extensions terminate as the food processes that lie on the outer layer of the basement membrane. The food processes of podocyte interdigitate to over or rather cover the basement membrane. The interdigitating processes are separated by gaps, that is the space between two adjacent food processes, which are called as slit pores or filtration slits and having a width of 20 nanometer. The filtration slits are bridged by a thin uh, diaphragm called as filtration slit diaphragm that contains small rectangular pores which do not allow free passage of the molecules. The filtration slit diaphragm is made up of a key molecule which is called as nephrin. The nephrin molecules form a zipper-like structure. The minute rectangular pores with the dimension of about 40 into 140 angstroms are formed between the prongs of the zipper. Thus, filtration, especially the protein molecules, is greatly restricted by these nephrins. Filtration slit diaphragm also contains negatively charged particles that prevent passage of proteins. So, so this is the glomerulus, and you can see the filtration barrier, which is mainly made up of endothelial cells, basement membrane, and the food processes of the podocyte. Okay, and in between the food processes, I mean there the filtration is going to occur. Okay, so these pores are called as filtration slits. This is another diagram, the podocyte showing the primary processes and the secondary processes. Okay, so and the space between these, they are going to allow the filtration, and which are called as the slit diaphragms. As we understand from the anatomy of filtration barrier, filtration through the glomerulocapsular filtration barrier occurs mostly through the extracellular route that consists of pores in the capillary endothelial cell layer, basement membrane, and slit pores in the food processes of podocytes of Bowman's capsule epithelium. The molecules with effective molecular radius 20 to 40 angstrom and cationic molecules are easily filtered through the filtration barrier. In diseases like nephrotic syndrome, the permeability of the glomerular capillaries to protein increases, and that is going to lead to proteinuria. Mesangium. This is an important component of renal corpuscle, and it contains of mesangial cells and mesangial matrix. Mesangial cells are derived from the blood monocytes and are the components of mononuclear phagocyte system. Mesangial cells are present in the mesangium close to the glomerular capillaries. The function of the mesangial cells are they provide structural support to the glomerular capillaries, they secrete extracellular matrix, they secrete prostaglandins and cytokines, they're phagocytic in nature and they kill and remove the foreign organisms and debris in the kidney by phagocytosis. The mesangial cells contain contractile units that are present in close contact with the glomerular capillaries. Therefore, the contraction and relaxation of the mesangial cells influence alteration of GFR by changing blood flow through glomerular capillaries or by changing capillary surface area. Mesangial cells that present outside the glomerulus, they are called as extraglomerular mesangial cells, and they are also called as Lassie cells or Gour-Martin cells. They are mainly located between afferent and efferent arterioles. They secrete some quantity of renin and erythropoietin and also possess phagocytic activity. Mesangial cells are involved in the development of immune complex mediated glomerular disease. Okay, so this diagram shows the location of the mesangial cells. Okay, so where you can see the nucleus of the endothelial cells, and these are the mesangial cells. Coming to juxtaglomerular apparatus, this is, if you see the structure, the thin ascending limb of the loop of Henle when comes in contact with the glomerulus of the same renal corpuscle, the structural modification occurs in the tubule and afferent and efferent arterioles. The entire modified structure, tubular and vascular components and cells between them is called as juxtaglomerular apparatus. The structure includes mainly the macula densa of the thick ascending limb of the loop of Henle, juxtaglomerular cells, that is the modified muscle cells that mainly from the afferent arterioles, and the Lassie cells, which are extraglomerular mesangial cells. Okay. So when thick ascending loop of Henle, when it come in contact with the same renal corpuscle, so they get modified, and with the afferent arteriole of the glomerulus and with the mesangial cells, they are going to form the juxtaglomerular apparatus. Macula densa cells are modified epithelial cells of the thick ascending limb of the loop of Henle when it comes in contact with the afferent and efferent arterioles. The terminal portion of the thick ascending limb of loop of Henle that passes through the angle formed by afferent and efferent arterioles of the same nephron processes this tubular epithelial modification, and this appearance is a dense area, and hence they are called as macula densa in the tubule, as epithelial cells are densely packed in this region. Macula densa acts as a sensor that monitors the change in the ionic composition and rate of flow of tubular fluid in the lumen of the tubule. It provides appropriate feedback signals to renal corpuscle to change the rate of filtration so as to meet the need of kidney function. Therefore, it is an important component in the tubuloglomerular feedback circuit. So this is the diagram of juxtaglomerular apparatus. Here you can see the macula densa, which is a modified part of the distal convoluted tubule, and here you can see the afferent and efferent vessels, where some of the cells they get modified into the juxtaglomerular cells, and these are the mesangial cells. The extraglomerular mesangial cells are also which are called as Lassus cells. Here you can see the podocyte, Bowman's capsule, and interglomerular mesangial cells in and around the glomerular capillaries. Juxtaglomerular cells are the granular epithelioid cells that present mainly in the tunica media of the afferent arteriole. Few are also present in the efferent arteriole that come in contact with the terminal part of the thick ascending limb of loop of Henle. In fact, these granular cells are modified vascular smooth muscles within an epithelioid with an epithelioid appearance. The cells are highly granular as they contain many secretory granules. They are also called as Pulkin's cells. Juxtaglomerular cells secrete renin that activates renin-angiotensin system. Lassus cells are present in the triangular space formed by efferent and afferent arterioles in the macula densa. Lassus cells are the mesangial cells that present outside the glomerulus and hence called extraglomerular mesangial cells. These are granular cells that secrete some quantity of renin and also erythropoietin. See the function of juxtaglomerular apparatus. They secrete renin and activates the renin-angiotensin system, which is involved in primarily the regulation of blood volume and the blood pressure. Macula densa cells acts as a sensor that detects the change in the rate of flow and the volume of flow in tubule and the composition of the tubular fluid. Lassus cells secrete renin and erythropoietin. Renin-angiotensin system. So renin is an acid protease secreted from the JG cells of kidney. So this glycoprotein having a molecular weight around 37,000. Like other peptide hormones, it is synthesized as a preprorenin and that finally becomes renin. And renin acts as an enzyme to convert angiotensinogen to angiotensin 1. Renin activates renin-angiotensin system, which is essential for regulation of ECF volume, and blood pressure. These are some of the factors which are going to increase the renin secretion and decrease the renin secretion. So these are the factors which are going to increase are the decrease in the blood volume or pressure, decreased plasma sodium concentration, and increased potassium concentration, increased sympathetic activity, increased circulating catecholamines, prostaglandins will increase the renin secretion, whereas increased plasma sodium concentration, increased blood pressure, ADH, and angiotensin 2, they're going to decrease renin secretion. Conditions in which renin secretion is high are in hypotension, hyponatremia, hyperkalemia, hypovolemia, dehydration, constriction of the renal artery, cardiac failure during standing, cirrhosis of the liver, and psychological depression. The role of sodium in renin secretion. Sodium is an important electrolyte that controls renin secretion. Decreased plasma sodium decreases the filtration of sodium through the renal corpuscle, that is through the glomerular filtrating membrane, and this decreases sodium concentration in the tubular fluid. The decreased sodium load is sensed by the cells of macula densa, which immediately provides feedback information to the JG cells to secrete renin. Renin activates renin-angiotensin system that results in angiotensin 2 formation, and angiotensin 2 increases sodium and water reabsorption that restores ECF volume. The opposite mechanism operates when sodium concentration increases in the plasma. Angiotensinogen is a glycoprotein synthesized in the liver, and the concentration of angiotensinogen in the plasma increases by the following hormones like glucocorticoids, thyroid, estrogen, cytokines, and angiotensin 2. Angiotensin 2 provides a positive feedback for further synthesis of angiotensinogen. Angiotensinogen is a physiologically inactive. It acts as a precursor of the formation of angiotensin 1. Angiotensin 1 is converted to angiotensin 2 by angiotensin converting enzyme. So this enzyme is a carboxypeptidase, acts on the angiotensin 1 to convert into angiotensin 2. Though the ACE is synthesized in and secreted by the endothelial cells of the blood vessels throughout the body, it is more formed in the blood vessels of lung. Hence, the major conversion of angiotensin 1 to 2 occurs in the lungs. It is going to exist in two forms, somatic and general form. However, physiological significance of existence of two forms is not known. So ACE, that is angiotensin converting enzyme, is also known to inactivate bradykinin. Angiotensin 2 formed from angiotensin 1 by AC. Angiotensin 2 has a short plasma half-life of 1 to 2 minutes, as it is rapidly metabolized by various peptidases and is rapidly converted uh, to form angiotensin 3, and angiotensin 3 is converted to angiotensin 4. Physiologically, angiotensin 2 is the most active angiotensin, and angiotensin 3 and 4 also physiologically active but not effective as angiotensin 2. Plasma renin concentration is measured to determine the angiotensin activity in the body. Physiological actions of angiotensins. 1 acts as a precursor of angiotensin 2, and 2 is the most important among all angiotensins. All of its actions are aimed at increasing the blood volume and pressure. Thus, the function of angiotensin can be divided into central and peripheral functions. Peripherally, it's one of the most potent vasoconstrictor. The vasoconstriction potency is about 8 times more than that of norepinephrine. Thus, when injected, it increases the blood pressure drastically. However, the vasoconstrictor activity of angiotensin 2 decreases in cirrhosis of liver and in patients with hyponatremia. A chronic increase in the angiotensin 2 level in the plasma in these conditions decreases the angiotensin 2 receptors on the smooth muscle of the blood vessels. Therefore, vasopressor activity of angiotensin 2 is less in hepatic cirrhosis and hyponatremic conditions. It increases the synthesis of secretion of aldosterone from the adrenal cortex. In fact, angiotensin 2 is a major regulator of aldosterone secretion. Therefore, renin-angiotensin system is called as renin-angiotensin-aldosterone axis. Aldosterone increases sodium water reabsorption from kidney that increases the ECF volume. Angiotensin 2 directly increases release of norepinephrine from postganglionic sympathetic neurons. Thus, it also potentiates the vasoconstriction through norepinephrine release. Angiotensin 2 causes contraction of the mesangial cells that decreases the glomerular filtration. Therefore, angiotensin 2 decreases the GFR. Angiotensin 2 increases sodium and water reabsorption from proximal convoluted tubules of the kidney. The central actions, it acts on the brain centers to decrease the sensitivity of the Barrow's reflex. So this potentiates the pressure effect of angiotensin 2. It stimulates the thirst center on the brain to increase water intake. It does not penetrate the blood-brain barrier. However, it acts on the circumventricular organs. These are the structures present outside the blood-brain barrier that include subformational organum vasculosum of lamina terminalis, that is OVLT, area prostrima, and the posterior pituitary. The subformational organ and the OVLT are almost the third center of the brain. Angiotensin 2 increases secretion of ADH from posterior pituitary. It increases the secretion of ACTH from anterior pituitary. Another function is it acts as a neurotransmitter in some of the areas of the brain. Angiotensin 3 possesses about 40% of the vasopressor activity of angiotensin 2. However, it has 100% aldosterone secreting activity of angiotensin 2. Thus, angiotensin 2 and 3 are involved in aldosterone secretion, whereas angiotensin 2 in the primary it is primarily involved in the regulation of blood volume and the pressure. Angiotensin 5 is the recently discovered angiotensin. It has some biological activities in the brain. Probably it stimulates vasomotor center. Angiotensin 2 receptors, there are two types of angiotensin 2 receptors, that is 81 and 82. 81 receptors present in the arteries and adrenal cortex. Acting on 81 receptors, angiotensin increases the intracellular calcium level via phospholipase C that forms the DAG and IP3. So there are two subtypes of 81 receptors, 81A, 81B. 81A receptors are present in the blood vessels and brain, and 81B receptors are present in the adrenal cortex and anterior pituitary. Whereas 82 receptors, they are attached to the G proteins that increase intracellular cyclic GMP. These receptors are mainly present in the fetuses and neonates. However, they are also present in the brains in the adults. Okay. So this is how the renin-angiotensin get activated. The angiotensin gets converted into angiotensin by the action of renin, gets converted into angiotensin 2 in the lung by the action of angiotensin converting enzyme. So then the angiotensin 2 is a potent vasoconstrictor. It increases the aldosterone secretion, and hence it is going to restore the blood volume. Whereas the angiotensin 3 and 4 have the same function, but they're less potent. Clinical importance of reticular renin-angiotensin system. It is implicated in the genesis of hypertension. Renin increases the angiotensin formation that causes intense vasoconstriction, which in turn increases blood pressure. Therefore, the conditions in which renin secretion is high, blood pressure is invariably high. This is important physiological basis of renal hypertension. In experimental animals, hypertension is produced by constricting the renal artery, that is producing renal ischemia, and this is called as Goldblatt hypertension. Etiologically, hypertension is classified into three categories depending on the concentration of renin in plasma: hyperrenin hypertension, normorenin hypertension, and hyporenin hypertension. In the treatment of hypertension, the drugs that inhibit ACE, that is which are called as ACE blockers, are very effective in controlling blood pressure than other group of drugs. The commonly administered ACE blockers are captopril and enalapril. Angiotensin 2 receptor blockers like losartans have been recently tried. There is a tissue renin-angiotensin system as well. In many tissues, there is a RAS that generates angiotensin 2 independent of plasma angiotensin 2. They are also found in blood vessels, uterus, placenta, exocrine, pancreas, heart, adipose tissue, anterior pituitary, and in some parts of the brain. And the exact function of a tissue RAS is not known. See the innervation of the kidney. The renal nerve controls renal blood flow, glomerular filtration rate, and electrolyte and water reabsorption from the body. The renal nerve contains sympathetic noradrenergic fibers, and there is no parasympathetic innervation to the kidney. Sympathetic fibers are also secrete dopamine. Stimulation of sympathetic fibers causes vasoconstriction of both afferent and efferent arterioles. However, afferent arteriole constriction is more, and this decreases the renal blood flow and glomerular filtration rate. The JG cells are innervated by the sympathetic fibers, and activation of sympathetic fibers results in increase renin secretion. So this is in brief about the introduction to the renal system. Thank you.